All right, here we go. So first of all, autism is a neurodevelopmental disorder that we know is characterized by deficits in social communication and social interaction, and kids with autism and adults with autism also might have restricted and repetitive behaviors and interests. But there really is a range of severity and support needs represented in autism. I use the term sort of autism and autism spectrum disorder really interchangeably. I represent and understand that all individuals may have a real range of severity and support needs that we're reflecting here today and who might be interested in the work that we're going to present. What we do know, though, taking that into account and sort of current diagnostic criteria, is that according to the Autism Developmental Disabilities and Monitoring Network, in 2022, 1 in 31 8-year-olds in the United States were identified with autism. We also know that we seem to be doing a better job of diagnosing autism earlier in life. And so that's the top graph you can see on this page here, where we can see that among children with autism, those born in 2018 were 1.7 times as likely as children born in 2014 to have been identified as having autism by 4 years of age. So while we can think about this as a real change in prevalence and a change over time, one of the other things that I like to say, putting my public health hat on, in some ways means that we are doing a really good job of increasing awareness and thinking about improving screening and recognition of persons with autism to try to help them get supports and the services they need to have the best lives they possibly can. And so in some way, these changing numbers are something that actually reflects good public health practices and services in place. But when we think about autism, much of the attention lately has come from the direction of what causes autism. What are the underlying factors that contribute to it? And my talk today is going to address both genetic factors and environmental factors and how we work to study them together. Starting from the perspective of genetics, we know that many neurodevelopmental disorders are influenced by genetic factors and that they run in families. So twin studies tell us that autism, but also things like ADHD and cognition and measures of IQ are perhaps influenced by genes, or they are heritable, they travel in families. And specifically for autism, we know that it's more common in full siblings of persons with an autism diagnosis as well. This figure here nicely lets us see that individuals with a sibling with autism are more likely to have a diagnosis of autism themselves, about a 7 to 8-fold increase over what we might see in the general population. But what is often sometimes more underappreciated is the fact that many distributions that would fall along a spectrum, things that we call traits, are also shifted in siblings of persons with autism. So the Mullen Scales of Early Learning is a measure of overall cognitive ability, and we often find shifts in those Mullen scores on average among siblings of persons with autism. Similarly, the Vineland Adaptive Behavior Scale is one of our standardized tools to look at overall functioning in daily life and broad sort of impairment and ability to live independently. And we find oftentimes that more siblings of individuals diagnosed with autism might be more delayed on this scale. They might have a harder time doing some of those independent daily tasks. And finally, an assessment called the Social Responsiveness Scale, which really measures social communication across the spectrum. We see that to be shifted. Those distributions look a little different amongst individuals who have cases of autism in their families versus not. And they actually look even a little different than people who might have sort of a non-specific language disorder as well. So this tells us that by studying siblings, we might be able to get insights not only to autism as a diagnosis, but the trait part of the spectrum that we see represented that lets us think about heterogeneity and differences among people. And it's also important to start to point out when we're thinking about factors that could contribute to autism or broader traits of autism, that sometimes genetics might influence the odds of a diagnosis, and that's sort of where I started from here, but they also might influence severity. So this picture here comes from a study of identical twins, and what we see is that their social responsiveness scale scores that measure overall social communication ability don't perfectly align, even though theoretically they share all of their genes, right? So, and those kids both had autism. So what would contribute to this difference in presentation? And wonder what are some of the possible explanations that we could have for the fact that genetics don't seem to be a perfect predictor either of diagnosis or of severity of sort of impairment across the spectrum. So one of the things that we study in public health is the environment. And so We could think about chemicals specifically. This picture here certainly looks at specific chemicals that might be hypothesized to be neurotoxicants and how they impact development and programming in the brain. And then as a person ages, then perhaps those cells don't mature they should. And then that might result in neurological changes or other changes downstream that could present with a given disorder. And part of the reason that we think about the environment that's really attractive for us in public health is it's something that we can try to modify. It's a possible target for intervention. So the environment can include anything from chemical factors out there in the air or in the water or in the food to social factors, to psychosocial stress and access to care, to lifestyle factors. So things like alcohol use or smoking use, or your broader ecosystems around you. What kind of neighborhood you live in? Is it walkable? What's the temperature outside? And the epidemiology part of environmental epidemiology lets us think about how health broadly comes together. It can tell us how common some of these environmental factors might be, and it can also tell us how big of an impact it might have on a population overall, because the reason we care about those population impacts, right, is that's how we impact policy change, how we get people interested. Finally, these environmental studies can help us think about mechanisms and pathways. So, genetics research is always great in autism because it helps us think about mechanism, helps us think about pathways in the brain that might be impacted. What if we could find environmental factors that helped us tease apart some of that specificity around maybe severity and functioning or language development and how that's different in persons who are verbal versus nonverbal who all have the same autism diagnosis? We can also think about biomarkers of these exposures, which could be really interesting to study too if we think about overall burden. So if the environment is sort of the overall impact of all the circumstances around us, anything that can impact growth or health or progress of functioning of someone or something, we have now tried to develop tools called the exposome that let us think about measuring all of the exposures a person would have in their life and how those might then relate to health. So that exposome concept is really important because it lets us move from sort of the abstract idea of the environment into how we actually measure it. So how do we measure the environment in population-based studies? So as many of you have participated in Spark, I'm certain that you've completed lots of questionnaires. So we ask people lots of questionnaires, right? That's our first line of attack here to try to think about how to measure the environment. We ask questions about medical history during pregnancy. We ask questions about what happened during birth. We ask questions about, what's in your home? What year was it built? What did you do for your job? What sort of things were exposed to? Where did you live? And those questionnaires help us really begin to get a baseline information of a broad range of things that fit into all of these different environmental categories. We also think about linking electronic health records. So increasingly, more and more health records across systems in the United States are electronic. And if we can make those linkages between family members within health systems, that can be incredible data for resources in investigation and population health. Additionally, based on an address, we can then look at a latitude and longitude, which then we can use a GPS measure, much like any of the data you might lose to locate a direction or a map where you're going in a car or on a phone, to think about linkages to geographic or external environmental factors. So things like air quality or water quality or neighborhood safety indices or access to primary care physicians, right? It might be in a given area. That lets us think about proximity and access and environmental toxicants. And finally, sometimes in our population studies, we collect biomarker measures of given exposures. So exactly from blood or saliva or hair or baby teeth or toenails, we can measure basically deposition of particular compounds. And our measurements are getting better and better that let us move towards this exposome idea, that let us integrate these different types of data sources to think about overall how people are exposed to. How does your chemical environment interact with your social support factors, interact with your access to care? And if we can layer genetics on top of that, perhaps then we really do begin to study more mechanisms that might lead to autism, but also impact functioning and quality of life among persons and their families with the diagnosis. So what have we sort of studied right now out there in the literature? What is sort of a short list of things that we have seen out there in terms of these modifiable potential environmental factors? So one bucket of them is toxicants. So things like air quality and pesticides and phthalates exposure. We think about demographic factors. There's some debate as whether or not those are truly modifiable, but spacing between births, individuals' ages, which can also be connected to genetic factors, and then complications that might happen during pregnancy. So things like prenatal vitamin use or infections or fever or diabetes or BMI. These are all sorts of things that we're interested in because we know that they have effects on the health of the pregnant person and of the child. So what is the state of the literature for some of these factors? So first off, maternal infection during pregnancy has been highly replicated across many studies. We can see the consistency of effects here in this picture, which nicely summarizes an association of about 1.3 across 36 population studies. So that's when we in public health start to think, oh, this is a great convergence of evidence. These effects go largely in the same way, and they're telling us a similar story. Similarly, fever during pregnancy has been associated pretty consistently with an increased odds of autism across methodologies, across types of diagnoses, across those sorts of studies. Folic acid has been of many interest, largely because we know that folic acid and fortification there has helped decrease the risk of neural tube defects. It has been evaluated in a couple different population-based studies. This one compares people with autism to people with typical development, and looked at the amount of folic acid intake actually that the mother had prior to pregnancy, in association with odds of an autism spectrum disorder diagnosis in a child, and actually found that having taken folic acid prior to pregnancy, so when they were planning a pregnancy, actually perhaps could be protective. In other studies that have looked at folic acid and more specifically just any prenatal vitamin use in the period before pregnancy among younger siblings of a child with autism, so working in this background family's situation here, has actually also been shown to help be productive. So perhaps it's something specific about folate, or perhaps it's because women who take a prenatal vitamin and have that higher folate intake might be more likely to have better prenatal care and then more monitoring moving forward. Finally, one last example here comes from Norway, where we looked at data. They looked at data of whether it's folic acid or multivitamin supplements before, during pregnancy, specifically looking to see if women who took them before pregnancy or during pregnancy had decreased odds of having a child with autism, and it seems that they both do here. Shifting gears a little bit to other sorts of prenatal or perinatal type factors. Maternal conditions during pregnancy, and in particular metabolic conditions, have become increasing interest. So there have been a couple papers that have investigated the relationship between obesity and diabetes, and specifically pre-pregnancy body mass index or pre-pregnancy obesity status, jointly with gestational diabetes or pre-gestational diabetes, any diabetes here not connected to gestation. This data comes from a cohort uncollected in Boston, where they really see this joint impact or this joint effect of both obesity prior to pregnancy, as well as additional metabolic challenges coming from diabetes during pregnancy. And this is really interesting in some ways because obesity and diabetes are something that we monitor routinely during pregnancy. Perhaps this gives us a better target to think about continued follow-up of some persons who might be really vulnerable. Other data has let us think about perinatal factors and things like prenatal depression and associations with autism. So this manuscript here is neat because it's sort of pretty recent, just published about 2 years ago, and it comes from the Environmental Influences on Child Health Outcomes, or the ECHO program. ECHO was really novel in that it's an amalgamation of bringing together many populations that have independently done research across the United States and pulling them together as to one common study, so making it big and bold now. And really in our main analysis, we were able to look at almost 4,000 parent-child pairs to try to evaluate the relationship between depression during pregnancy and potentially having a child with autism. Here, you can see that overall, the odds of having a child with an autism diagnosis, and here specifically, more than diagnosis, elevated autism-related traits, so measured by that social responsiveness scale I talked about earlier that looks at social communication ability, was seen to be elevated in children of mothers who had a diagnosis of depression prior to pregnancy. And then in particular, that also seemed to be present among mothers who had more severe depression in that time period prior to their pregnancy. why do we care about this, right? Is that several studies have looked, for example, at selective serotonin reuptake inhibitors or other medications to try to help treat depression in relationship to autism. And this paper lets us really begin to study and to think about the fact that perhaps it isn't actually the treatment itself that might be indicative, it's the diagnosis itself. And part of the reason of having that diagnosis could be due to shared factors that we know that happen etiologically in the brain across these conditions. I mentioned earlier that some of the sorts of things that we study are also things like environmental toxicants. There is a pretty robust literature around outdoor ambient air pollution exposure, so the air quality that we have outside us in the world, anything from particulate matter less than 2.5 microns in diameter or NO2. These are things that are routinely monitored around the United States outside. And this paper, further leveraging the ECHO consortia that I mentioned just now, actually looked even bigger at bigger data, right? So 8,000 mother-child pairs from across the United States, and overall found that there were significant relationships seen with increased presence of autistic traits measured by the social responsiveness scale again, and with diagnoses of autism, in relationship to higher air pollution exposure, and in particular exposures to PM 2.5 and ozone. What's sort of notable here, though, is that we saw differences in those associations in different regions of the US, because perhaps the underlying components are different, or perhaps the actual levels of exposure could be different, or they could be related to occupation. So further data and further a study needs us to understand why exactly these things are different around the world, around the country. And then finally, one more note about air pollution, because this is sort of an interesting pivot, is that there have been a couple papers also, and this one in particular, that have really tried to look at function. So perhaps we're not really thinking overall just about autism, right, as an outcome if we're looking at the relationship between air quality and brain health. Maybe we should be studying function. So how these kids do in their daily life. And does increasing air pollution exposure associate with poor overall function, even among people with autism, trying to get at representation of the full spectrum? And that's what this study shows here, is that increasing exposure to PM 2.5 in the early pregnancy was associated overall with poor performance on the Vineland Adaptive Behavior Scale, which measures sort of overall daily functioning and living skills. Now, it's important to point out that I said at the start of this talk that we are going to talk about genes and the environment together. And there has been this really neat analysis in this giant data set from Denmark, which can do such things in a unique manner, of over 1 million individuals and 18,000 persons who were diagnosed with autism from medical record. And what they tried to do is look at many of these factors that I just mentioned that we've done in smaller studies here in the United States, and looked at them one at a time, looked at them saying, well, what if people had multiple of these conditions? Because that's the way things really happen, right? You don't just have one health condition at any given point in time. There's not one characteristic that exists. And what if we looked at those comparisons between sibling pairs, where one sibling has autism and one doesn't? And the reason for that is because we started out this very talk by saying that we know that autism might run in families, that families that have one child with autism are more likely to have another child with autism. So perhaps it's that impact of the family factor or genetic factors working together with these other conditions that relate to health that could be most important. So what they find in this cool picture, and if you look all the way up at the top of the picture, you'll see those are the things that we find to be statistically significant, right? That really seem to be most associated with an autism diagnosis in this data from Denmark. And I have them listed here on the bottom. So these were things like personality disorders, major depressive disorder, and anxiety disorders. So 3 other mental health conditions that we know are biologically related to other mental health conditions. And then obesity, which we talked about in a couple slides ago, and then complications that happen during pregnancy. And so this is where it's interesting to say, wow, so these are 3 very different classes of conditions, and they obviously could all happen in one person together at the same time. But, because we're thinking about genes and the environment working together, how do these same factors that I just called out on this previous slide change if we look at them in terms of associations between a sibling with autism and a sibling without autism in the same family, trying to get at that genetic background? And so the 3 bars I have here highlighted in red shows some really interesting examples where our effect of a particular condition really changes whether or not we're looking at a sibling or not a sibling as a comparison. So the first one up there is asthma. So that's really interesting because it's something that goes from risk of autism to being protective when we look at siblings, right? So it totally changes direction. So that lets us think that it's something specific in the family that might be happening there, right? That gives us a signal that maybe it's not the asthma, it's asthma acting on this familial background that makes an impact. The next bar looks at major depressive disorder, whether it's recurrent, so more continuously happening, or just one single episode. So again, here we see this decrease in types of effects, but it's not as big as it was up there for asthma when we look within families versus across our whole population here. But we do see some decreases there. And those decreases we think are probably important because one of the things that we're looking at there is that, we know that genetic backgrounds for many psychiatric conditions and many traits are actually related as well. So that could explain some of those findings. And then on the bottom where is overweight and obesity, right? Because we talked about obesity and gestational diabetes. And again, we see this attenuation effects. So what I don't show you on this slide, because it got too busy and trying to make it explanatory to folks, is what happens if we look at fever and infection, right? Because that's a great question. And that's something that could happen in both pregnancies, multiple times if you were pregnant, depending on the time of the year. And in that comparison, we don't see this sibling difference. So that lets us think that maybe there could be something there that's happening independent of genetics. And so that is something that we want to try to explore further. So we take this sort of population data and studies in siblings and in families and try to move more towards studying genetics. And so for folks participating in SPARK or the Simon Simplex collection, we have collected a lot of genetic data to help us think about the biology of autism, right? What are the pathways that are changed in autism? What's important? There's a lot of different ways to measure genetics, as you can see in that bucket over there left on the slide, whether we're looking at common things or we're looking at more rare changes. I mentioned a whole bunch of different kinds of environment. And what we're trying to get at is, are we trying to look at statistical interactions or biological interactions, and how does this all come together? So how do we really bring it together? So some of the work that I've talked about has been in studies of hundreds of people, and some of it has been in thousands of people. So, is the bigger the better? Do we need thousands and thousands of people to try to do this work? Do we need lots of different kinds of expertise? Well, probably, right? Because if I'm an epidemiologist, which is what I am, I want to work with a clinician to help me get my outcomes right? And to really think about what could be impactful for folks. And so the type of information we gather across many different types of studies can vary in sort of the quality of depth, depth that we get. So if we're thinking about broad administrative data from healthcare systems or registries like they have in Denmark, we might have information on a lot of people, but the information that we might have about their health conditions, and in particular, autism, right? Whether it's a diagnosis or a continuous factor across a range of conditions or impairments, is more limited because all we'll have is just information on the diagnosis. And some other studies, like case control studies or cohort studies where we follow population over time, that maybe lets us ask more questions of folks, lets us ask some of these questions related to severity and function and a broader impact. And by continuing to do those measures over time, it lets us think about the life course of a person too. So we get very deep information. We get a lot of variety of information, but we're going to have to do it in a smaller set of people because it's really hard to do in a big data set. So to date, for better or for worse, there's been limited work published on gene-environment interactions specifically in autism. There's different types of genetics that have been studied, and there's a range of environmental factors that's studied. But overall, relative to sort of the broad literature of what we know about autism, this area hasn't gotten a lot of attention. Some of it, I think, is because bringing together this types of data has taken a long time. There are different types of genetic variation that we can see. But the most common that I'm going to talk about next leverages something called a genome-wide association study, where we're looking at individual change in the genome in a whole lot of different places, sort of agnostically, to discover where in the genome we think there could be changes. But what's important to take home here is the idea that common exposures and common genetic changes are easier to detect, and rare exposures and rare genetic changes are harder to detect. And what the truth is probably somewhere in the middle of all that, right? And trying to discover how it might go next. So I mentioned genome-wide association studies where we look across the genome. And so these pretty pictures here are called Manhattan plots because they're thought to represent and sort of look like the city of Manhattan, like a nice big city skyline. And there's a certain threshold that's drawn across all of these plots that let us say that these are where significant changes are in the genome associated with a given outcome. And so we have done these sorts of studies for all sorts of neurodevelopmental outcomes. This example here looks sort of at general cognitive function, a particular test called digit symbol, which measures executive function, and so higher level processing, ADHD, and then autism down there in the bottom corner. And it's that autism one that I'm going to show you some work that we have done here at Hopkins with many collaborators to try to take forward to see if we can make some sense in the idea of gene-environment interaction. So if we're taking from this genome-wide association study where we need these giant sample sizes and we have to do all these statistical tests, we have developed a tool called, with many other people have too, but we have applied it in this data set, called a polygenic score, where we can take each of those individual pieces of information and we can summarize them all into one single score. And the reason we like to do this is because then we can take that score and translate it into other populations where we might have smaller sample sizes, but that sort of really deep data about people's trajectories around functioning that we might have. And some of the places that we like to do this, probably no surprise, are in sibling cohort design. So following up over time, siblings of children with autism. Part of the reason this is great to do is it lets us really get that deep dive on what autism looks like in some of these kids and in these families. And it lets us think about sort of in utero or infancy starting points for these outcomes to measure. So we can think about what a child might look like at 6 months, or a child might look like at 12 months, or what a child might look like at 36 months. And what's novel here is that the work I'm going to show you quickly here comes from 4 different sort of studies with these different designs that were each started for a different reason. Some of them were really meant to study early indicators of brain development. Others really were focused on early sort of detection methods that could be done in the clinic. Others were really focused on prenatal exposures. And sort of one of the things that we realized is that many of us had common data of this follow-up over time that lets us begin to try to think about how we could bring genetics together in a more powerful way. So here we can see overall, we pulled together these 4 different studies. We have 178 kids with autism in this data set of almost 900, 850 individuals overall. And we have a higher diagnostic rate of autism than would be present in the typical population because these are siblings of kids with autism again. And what we can see here, and Kelly Benke from our team here at Hopkins has really led this work, is that we look at the relationship between that autism polygenic score that I talked about coming from all this big genetic discovery research in relationship to autism. What we can see here is it does have some predictive ability. So across all the cohorts, that odds ratio is about 1.2. So it's saying that there's about a 20% increased odds of having an autism diagnosis as your score gets higher. But obviously, that's not a perfect predictor, right? But one of the other things that we have learned and really begun to think more and more about is, can these genetic factors that might be related to autism also tell us more about functioning across the spectrum? And so here we look at the relationship of that autism polygenic score in relationship with the overall scale of the Mullen scale of early learning. So the Mullen here is meant to measure sort of like overall cognitive ability. And what we see here overall when we look across our data sets is there isn't anything really consistent, right? It doesn't look totally straightforward across our studies, across that relationship between increasing autism, common genetic risk genes, and the Mullen. However, what gets really interesting is if we look within kids' later diagnosis at 36 months, so whether these siblings of children with autism were diagnosed with autism, some other state of non-typical development, typically indicated by a lower cognitive score, or typical development. And what we see here now is that having a higher threshold or having more autism genetic liability coming from this common variant score is really associated with poorer performance on this cognitive test. So they do worse overall in terms of the early learning composite, which is sort of the overall scale score that we see, and have poorer language development. So both expressive and receptive language, as well as fine motor development as well, among siblings of kids with autism who weren't diagnosed with autism at all, who really thought to have typical development. So this was interesting to us, right? And that we can really try to get at how can we use some of these big tools in autism research to think broadly about impacts of development and functioning on other kids and families and related outcomes as well. So one of the things important to mention here, right, is this analysis was done in about 850 kids across 4 studies. So if we really want to move gene-environment interaction to a bigger scale, we realized that we needed a network. We needed bigger data and we needed better collaboration with great researchers and smart people. And so that is where our network on gene-environment interaction, or the GEARS network, was born and came together. Christine Ladacosta there, who's also here at Hopkins, is my co-lead on the network. And what we have done is really built an infrastructure for trying to develop gene-environment interaction studies on a broad scale. So there are 18 individual study sites. You can see Spark is one of those in there. And together across all of these 18 studies, it compiles about 175,000 individual participants. We have worked with folks who actually develop brain organoids or model cellular systems to try to think more about the biology of our work. And most importantly, perhaps, in trying to do this work around gene-environment interaction and autism, we have partnered with our community advisory board and the Wendy Clegg Center here at Johns Hopkins to really think about what matters to persons with autism, their families, additional care providers, and other stakeholders. So the cool part here is that we have this big machinery set up now to study different types of genetics, really still largely focusing on that common variation part I talked about earlier, and different types of environments. But all of our cohorts have information on autism diagnosis, which is great. But we can move beyond that because we have so many folks involved in different types of study designs that we can sort of look at severity of autism symptoms or verbal ability, or perhaps some of these broader quantitative traits like the SRS that I mentioned earlier, looking at social communication. And then finally, where we think actually might end up being most impactful to folks in the autism-related community is to be able to look at how genes and the environment work together to impact co-occurring conditions. So perhaps it's a particular combination of environments and genes that really impact, sleep health among persons with autism, or perhaps other mental health conditions that we could evaluate. I mentioned that we've worked with folks who have a brain organoid model system. I'm not going to go too much into that today. I think it's important for translation that lets us help us think about biology in ways that we really can't in population studies, but it's not my area truly of expertise. And then I mentioned the last part that I said was really important is thinking about dissemination and outreach of our work. How do you talk about science in a way to the community, to stakeholders that really matter? How do we help make this information valuable? How do we think about really getting input put in place for folks so that we can think about bidirectional communication and who might be most impacted and use this information? So overall, I would say right now we have our network up and running, which is pretty awesome. We've got things up and moving. We can tell now that our cohorts that have been part of our network span a huge range of birth years, which is really exciting if we're thinking about sort of trends in time in terms of environmental exposures or environmental toxicants. So many of these cohorts were actually started in the late 1990s and followed kids up into the last few years. There are different places all over the United States and different types of sample sizes and different study designs. We do have that genome-wide association data I measured that helps characterize common variation available in all of our study participants, which is exciting. But we only have a couple that have data on rare variation. I know many genetic studies and repositories have really focused on that. So that's something we hope to do a better job of. In terms of those environmental exposures, we have all sorts of data, anything ranging from those questionnaires to geographic linkages to biomarkers to health records information, anything from behavioral data to health conditions during pregnancy to toxic chemicals. And some of those sample sizes can let us study these things in maybe as many as 70,000 participants. And then, as I mentioned, there are many outcomes to study. So everybody has information in our network on an autism diagnosis. But we are really fortunate to have as many as 10 to 20,000 individuals where we could really look at severity of measures, right? Or particular domains of traits or language development or social communication to try to understand, is there a threshold upon which these things really seem to be impacted by combinations of genetics and the environment together? And then finally, I mentioned those co-occurring conditions because they might provide targets for real public health impact and improvement as well. So what have we found so far and what have we done in terms of some of the actual science? So our first analysis that we pulled together has looked at autism diagnosis as an outcome, and fever and infection as the environmental exposure. And the reason for that is because it's been replicated in all those studies that I showed you previously, and also because many of our cohorts had that data available. In terms of genetic factors, we did look at that autism polygenic score, so sort of that increasing autism genetic risk. And then we look at the genes times the environment, like the joint effect of them together, right? And so it's really excited that this work was presented at the International Society for Psychiatric Genetics a few weeks ago. And overall here in our data, we do still see consistency in terms of prediction in autism diagnosis in relationship to the autism polygenic score that I mentioned earlier, coming from just one or two of these individual populations. Again, it's not a perfect predictor, but it is something that lets us see that our science is converging. We are taking data from these large discovery data sets where we're trying to find individual markers of genes that are associated with autism diagnosis, and we can effectively summarize them and transport them to other populations where they come together and still have that same impact. We looked at infection reported during pregnancy in relationship to autism diagnosis. And here you can see overall that during pregnancy, the odds of having a child with an autism diagnosis, if you have an infection of any kind, sort of grossly reported here during pregnancy, it's about 1.6. So that's a pretty consistent effect size that we see across many of these studies and consistent with some of the other data that I showed you. And similarly, fever, the effect is a little bit smaller, but again, really consistent across all of our studies. So we can see independent effects of genes and independence of fever, effects of fever and infection. But we don't see any of these joint effects when we look at them multiplied together. So this lets us believe right now that perhaps these two factors really are operating independently, that it doesn't think of it, it isn't a result of sort of familial genetics or inherited factors that are coming together. Maybe they have different mechanisms that we need to be studying and thinking about. So I mentioned dissemination and outreach is an important part of our network as well. And some of the things that we wanted to talk with folks about are, how do you want to receive your information? What does the word environment mean to you? How do you think about the role of, research in autism, and in particular genetic research together? And so our team really nicely pulled together a focus group, series of focus groups across multiple stakeholders to try to answer this set of questions. So what are the priorities of stakeholders amongst adults on the spectrum, parents of caregivers of individuals on the autism spectrum, providers of individuals, and siblings? How can we give research back to the community? How can we help disseminate it to have it make sense? What are some of the barriers to contributing? And Michelle Trice and Jessica Walton, whom I don't have a nice picture for, I need to get one for her, so she's missing there, did this work and also recently presented at the International Society for Psychiatric Genetics meeting. And so they conducted these 4 groups amongst the stakeholders I just described and had 60 to 90-minute virtual sessions that had a maximum of 6 participants over the period of about 6 months. We sampled individuals, anybody who wanted to participate across the US, and we tried to ask many open-ended questions about many of these factors that I just described, how we think about including folks in research and how they want to get information. So 19 participants participated in our study, different types of stakeholders. 5 were caregivers, 6 were adults with autism, 5 were siblings, adult siblings of people on the spectrum, and 3 were medical providers across the US. So, our first question we talked about is, what do you think about the role of genetics in autism? And you can see here across all of our groups, people largely said, yeah, there's something about genetics that seems to be important. And 3 of those groups said, yeah, that autism seems to be inherited in families, right? But it was really interesting to see where some of these things that look to be different. So, autistic adults voiced that they thought their brains maybe were really different on a genetic level, or caregivers really thought that they think there's strongly an environmental component to autism. In terms of the research priorities of stakeholders, it was fascinating to see that across all of the groups, everybody really wanted us to prioritize, how do we deal with the more troublesome symptoms of autism or co-occurring conditions? How do we try to work and the reason to do that, right, would be to prioritize need for support or access to services. How could genetic research help people with autism today? How can we help get at those things that might be most deleterious or debilitating, right? 3 of the 4 groups, so autistic adults, caregivers, and siblings, also really were advocating for research that leads to timely diagnosis, factor access to services, and help differentiating between, is it autism or is it something else, right, that's happening in my person, because that matters how we might think about treating for services. And then the group also had some specific themes, like, Perhaps siblings of individuals with autism wanted to understand how genetic research could help them understand they might also have a sibling with autism, or could we use this to help understand the basic biology of autism and help it be then thusly destigmatizing for the disorder, or people who identify as on a spectrum. Some of the specific things that folks said, had to deal with the idea of, thinking about blame, which is something we try to be very careful not to do in any of our research. We think about trying to understand, the percentage that corresponds, as it says there, to genetic versus environmental factors. How do we help people have better, healthier lives and the best lives that they can? Some of the barriers that were cited across the board had to deal with mistrust in information or ease of participation. We realized that we need to make it easy for folks to participate in these research studies. And both autistic adults, caregivers, and providers, also again mentioned this idea of mistrust and misinformation, and so making it difficult that we really didn't know what to believe. Caregivers nicely also pointed out that perhaps concerns around privacy that might come into play as well in genetic research. One of the things that we were most excited to hear from our focus groups about was really how we could disseminate research information. What is the best way to talk about science, and how do we get it out there so that it really can impact people? And so it was great to see across the board, everyone wanted to see this information come from trusted, reputable sources. We wanted it to be lay friendly and diverse. More sources of information coming from voices specifically from the autism community, which is a really exciting point to see. And different types of dissemination, anything from social media to infographics to podcasts to newsletters, moving beyond sort of typical academic literature. So what have we learned so far, right, in our network, and how does this all come together broadly in this data of gene-environment interaction? So, our limited work so far tells us this idea of a polygenic score makes sense. It's consistent across studies. And it's a nice example, I think, of how we can take information from one type of study and move it to different collaborations to help us understand more about autism biology. We also can see here too that fever and infection, based on our work from our network, do really seem to be statistically significant and associated with autism, but the environment and gene findings together look like these factors are independent. So what are we going to do next in terms of many of our analyses for our gene environment network? We're going to change our outcome. We want to look now more broadly at social communication deficits across the spectrum and at cognitive function, and in terms of our outcomes, to try to get a better handle on broader representation. We also want to look at maternal metabolic conditions, so digging a little bit more into some of those findings that I mentioned from earlier studies around BMI and diabetes, and air pollution as a potential modifiable biotoxicant. But some of the big lessons that we've learned about how important some of this work is, can really have to do with thinking about reproducibility, but most important, thinking about how we can clearly transmit our information to stakeholders, and then the ease of participation. So how can we make it easy for folks to participate in things like focus groups or broader discovery studies, like we do it in our work, or like what Spark might be? So what's next, right, in terms of gene-environment interaction? So these big scale studies like the GEARS network that require multi-site and interdisciplinary folks, I think are really what are going to drive the future. There has been a whole host of awards recently put out there specifically to expand gene environment interaction and research and environmental research in autism. It's exciting to see these come out, that we won't be the only game in town anymore, that there'll be many more folks who are working in this area to let us really expand and replicate, to really begin to believe, are there specifically types of genetic factors we should be studying? How do we work this together to translate impacts around messaging for persons with autism and their families? That additional expansion is really essential to make that happen, and it's really exciting to see that coming to the forefront soon. And so with that, I thank you all so much for your time and the opportunity to talk today. I'd like to always recognize the members of my team, many of them you can see there, and our networks broadly together, and the folks that have supported our work. Thank you so much.
Thank you so much, Dr. Volk. This was really interesting. I know a couple years ago you did a talk for us, and I think this, for those who haven't listened to that talk, I encourage you all to check that out on the Spark site. But this was a really nice sort of continuation of that. But so with the 10 minutes or so that we have, I guess we'll just jump right into Q&A. I'll gonna try and prioritize things that came in from multiple folks, just because obviously we've, if a few people are asking, I think it might be good to start there. So, um, you talked a bit about maternal factors, specifically pregnancy. We got a few questions from folks wondering about like, what about paternal considerations? Is there anything that we know about potential paternal factors that may impact
That is a
autism?
great question. So there is this huge lack in public health research about including dads in research. Nobody does it, and I have no idea why. It's something we need to do more and more. There have been a couple papers that have looked at things like paternal age. There are a couple of small studies that try to look at dad's occupation, for example. And then there's a couple papers by members of our team that have looked at sort of changes in factors that relate to gene expression and how those might relate to development of autistic traits, particularly in sperm. So that's relevant uniquely for fathers. But it's a great question. It's something that we have not done much about, and it's definitely an area of research that's more needed.
Great. Yeah, that's I didn't realize that that was lacking so much. Hopefully we can, in the future see some more there. And then additionally, you also spoke a bit about, and I think shared even a couple studies looking at siblings. I thought it was an interesting question from a couple folks. So if, scenario here, one sibling with autism, another does not have autism, when it comes to the genetics, is there likelihood or anything that we know about that sibling without autism potentially having children with autism? Hopefully I explained that correctly.
Yeah. So that's a great question. And specifically, that's not something that our team has looked at, right? So one of the things that I can do is point out the family studies, right? So risk of a sibling if they have it, or if a sibling have a diagnosis, if a sibling has a diagnosis of autism, right? How that works for them, sort of siblings of or children of siblings, I think is a further question under study. There are a couple efforts going on right now, one in particular funded by the Autism Science Foundation to try to next gen SIBS is what they're calling it. So like the next generation of siblings and children with autism who are now entering childbearing age. So hopefully soon we'll begin to have that better answers to that question.
Great, thank you. And then another question that came in from a few folks is there anything that we're learning from these gene environment interaction from all these studies that's helping us understand why there are more boys than girls that seem to be diagnosed? Is there anything there that you could tell us about?
So that is an awesome question. And one of the things that we're hoping to do sort of in our next round of analyses, one of the nice parts about working in this big collaborative framework is that we have enough data on girls with autism to really begin to understand that. Some of the folks that have looked at environmental main effects of particular chemicals is the best thing I can talk to, have really focused in on things that we noticed that may be endocrine disruptors. And so they think then perhaps those might have differential effects on girls and boys. But studies that bring those together with genetics have not yet been conducted.
Okay. Um, and then I think kind of in the beginning of your talk, I think you talked about a couple studies looking at different countries, outside of the US. Are there any trends that have been seen in sorry, I just want to make sure I'm getting to the crux of this question correctly because I thought it was interesting. Are there any trends that you've seen or cultural specific differences, from the United States or perhaps other countries in the environment that maybe are impacting or aligned with those like lower rates of autism in one country versus another? Again, hopefully I got to that person's question correctly. Yeah.
Yeah, so I think that what you're trying to get at, right, is to get the idea that some countries might have lower levels of whatever a particular factor is, right? And then they might also have lower rates of autism diagnoses, right? So one of the things that we want to try to be really careful to do is while we can look sort of like within a country as a whole or country to country, we don't want to extrapolate those comparisons of countries to people. It's this thing called the ecologic fallacy. So essentially what it means is saying a group equates to the person, right? So when we were trying to think about where to look at for some of these studies outside the US, I mentioned Denmark earlier and Sweden and all the Nordic countries have incredible data on things like conditions during pregnancy because they have these awesome health records that they look at for many of those studies, right? Other places around, and they also have some models that let us look at things like air quality or exposures or things like that. So those are some of the places that we're trying to look now and look to how our work in the United States really compares to what you see in other countries where that similar data could be in place. the catch is that because they have these awesome healthcare systems, right? How they diagnose people with autism and how they might become diagnosed or get subsequent treatment can be really different than what we see in the US. So it makes it really hard to tease apart, are these trends different across countries around the world? And are they really associated with lower rates of diagnosis? Or maybe we have better healthcare systems for some places. Maybe we're doing a better job of referring or treating or finding individuals. So it's a tough thing to tease apart, I guess, is what I'm trying to say, not very succinctly here. How we're getting the data and what systems are in place that relate to our given exposures or health conditions and subsequently diagnosis.
Great. And I think if we want to just, we're looking at that was kind of about like country to country, but if we want to maybe look come down a little, excuse me, come down a little bit and look at, like, let's just say in the US, for instance, something I think right at the top of your presentation, you talked about one of the examples of in your environment that can impact autism is just the walkability of
Right.
where you may live. So I think if we take that a little bit further, is like differences between, an urban environment where maybe you're walking more places, maybe there's public transit versus somewhere more rural where you're either relying on cars or things are further you know, s- you're getting around in just different ways just because of the sheer, the vastness of the space. Is there anything more you can say about sort of those kind of differences in environment, like different urban, suburban, rural in the States?
I mean, I can a little bit. So
Yeah.
specifically in the United States, right, one of the things that happens is we've looked, for example, at urban versus rural in relationship to autism. And not surprisingly, like it's more likely to have a diagnosis of autism if you live in an urban area. But why are you more likely to have that? Is it something due to your urban area, or is it because there are more healthcare providers with access who might or do refer to development and screening, right? And so that's a very specific example. And so when we think about these urban rural associations, or is it something about living in a city? Is it more chemical exposure, or but maybe it's increased walkability, and maybe that's good because it helps promote physical activity, or maybe it's higher stress levels depending on where you are. These factors based on geography make it really hard to tease apart with what we know about referral patterns and access to care, especially for an autism diagnosis in many parts of the country. So I think your question is right on and it's really important. Unfortunately, I completely don't have a satisfying answer at all as to, what it might be in a given geographic area because of it.
Right. That makes sense. And then, another topic you talked a lot about, I think we touched on this briefly. We were talking about, the maternal side of things, but specifically when it comes to pregnancy, quite a few people asked, do we know anything about, IVF pregnancy and autism? Is there anything there, or maybe not yet?
So there are a few studies that have tried to look specifically at IVF, and the tough part about this data, right, is you're looking at people who, for whatever reason, have had troubles with fertility already. So I would not change particular behavior based on the data that's already out there. I think that's something that we need to study more to really try to get at, is there something broadly related to difficulty maintaining and conceiving a pregnancy, more specifically in relationship to having any neurodevelopmental diagnosis.
Great, thank you. And then there's one country, excuse me, I'm reading
No
my notes
worries.
here, so I'm we're jumbled. But one question here that I just lost, but I think it's actually just really kind of a point blank. I like the way this person phrased it, and I can't find it. Oh gosh. Um, Oh, here we go. So I think for maybe somebody who perhaps joined late, maybe left early, is going to come back to this, maybe didn't even join us live today at all and is just going to go watch on their own. I think this was a great question to ask. So, is the general consensus that autism is caused by a combination of the interaction of genetics and environment?
Yeah, I think that is it, right? I
Yeah.
think that we know that autism has a genetic component, right? And that doesn't explain everything that we know about autism in terms of both diagnosis and in terms of variation across the spectrum. And so there has to be something else happening. And so pulling genetics and environment together moves us better, I think, towards thinking about what might explain that. It's not as simple as just one thing or the other.
Right. Thank you. I think that was probably just a general question that
Yeah,
maybe,
great.
yeah, like I said, if somebody joined late, maybe they're not sure or left early, they're not really sure where, so what is the takeaway here? And I think that was a nice succinct way to put that. And I think just one last question that I think is interesting, and I think again came from a few folks. Um, it goes back to pregnancy and the COVID. I think it's 2025. Obviously, we're the pandemic has passed, but COVID is still very present in our lives. Is there anything that we've learned or that scientists have learned about COVID and autism?
That's a great question. So actually, just like 3 weeks ago, there was a paper published that showed that women who had COVID infection while they were pregnant, they were more likely to have a child with autism, right? So one of the real questions that I would point out to folks, right, is that that makes total sense in sort of the broader literature of infection during pregnancy being something that might potentially change risk, right? Might increase the odds of having a child with autism. So it's totally consistent with everything else that we've seen with any other type of infection, right? And so this lets us believe that, so one of the things that needs to still be done moving forward, whether it's COVID or any infection, as we head into, cold and flu season here, right, is thinking about, is it just having any infection? Is it something to do with the immune system, right? What do we need to be doing there to have healthy pregnancies and healthy offspring, right? And, so there's more work to be done, but that's what we know right